Today, welcome to another physiology lecture. For endocrine physiology, the lectures are divided into five. The first three of these lectures will be discussed today, while the remaining two will be discussed next week. So our objectives for this set of lectures would be as follows. To review the basic principles of intercellular communication, especially the characteristics of hormones and receptors. And then later on in my second video presentation, we will be talking about the function and the structure of the hypothalamus and the pituitary glands. And lastly, in the third presentation, will be all about the thyroid gland, particularly the thyroid hormone, its synthesis, its release, and effects in other organs. So as I mentioned, this video presentation will cover the first objective. Unlike the other systems of the human body, the endocrine system is unique because it involves organs located in various locations. You'll have endocrine organs in the head and neck, in the abdomen, and in the pelvis. Okay. As what we have learned in the previous bimonthly, we already talked about the ovaries and the testes, their structure and function. In the next 2 weeks, you'll be learning about the other classical endocrine organs, such as the hypothalamus and the pituitary gland, the thyroid and parathyroid gland, a review on the parathyroid gland since you have that already when we talked about calcium homeostasis, the adrenal glands, and lastly, the endocrine part of the pancreas. But keep in mind that there are also organs which may have endocrine function. So these organs contain cells which can perform endocrine activities. For example, is the kidney, where erythropoietin is produced, and it has an endocrine function as it helps in the production of red blood cells. You also have some other organs such as the lungs and the heart, which contains cells which have endocrine function. So again, this is a figure from Guyton and Hall showing the different organs, endocrine organs of the body. And can you guess which of these organs have the following functions? Which organ is able to regulate sodium and water balance and control of blood volume and pressure? What do you think? How about an endocrine organ which regulates calcium and phosphate balance? Or the regulation of energy balance and control of fuel mobilization, utilization, and storage? Another is coordination of host hemodynamic and metabolic counter-regulatory responses to stress, which is the main organ involved. And lastly, there are also organs which regulate reproduction, development, growth, and senescence. Which organs do you think are involved in these? So I hope you'll be able to answer these questions during our Q&A session on Thursday morning. Before we talk about the hormones, let's have a review on the receptors. Keep in mind that there are basically two types of receptors. One which are found in the cell membrane, or what we call as your surface receptors, and the other one are the intracellular receptors. Remember that your surface receptors can either also be ligand-gated ion channels, or can also be receptors that span the transmembrane or the cell membrane, such as your G protein coupled receptors and your receptor protein tyrosine kinase, which is a catalytic receptor. You also have receptors found inside the cell. It can either be found in the cytoplasm or in the nucleus. Hormones can be classified based on the type of receptors they bind to. So those three types of receptors I mentioned, you have several examples here where your hormones would bind to. So for example, your TSH, ADH, and catecholamines would bind to G protein coupled receptors, while insulin and growth factor would bind to a catalytic receptor such as a protein tyrosine kinase. And of course, steroids and thyroid hormone would bind to intracellular receptors. We'll also talk about the characteristics of hormones, and foremost is that hormones use the bloodstream to carry messages to particular organs. Endocrine organs secrete their substances directly into the bloodstream. And the endocrine organs act as your first messengers. The mechanisms are stimulated by changes in the internal environment, and in this sense, the internal environment is the extracellular fluid compartment. And lastly, hormones are closely related to and functionally interdependent with the nervous system. A hormone can also be classified based on its chemical nature or where it is derived from. So basically, you have three classifications. You have proteins and polypeptides, amino acid derivatives, and steroid hormones. And listed on the right are examples of these classifications. So for proteins and polypeptides, generally they constitute the majority of hormones, and they may vary from 3 to 200 amino acid residues. The half-life can also be dependent on its nature. You'll have as little as 4 to 40 minutes for polypeptides, and it can reach up to 170 minutes for certain proteins. They are initially synthesized as pre-pro-hormones and undergo post-translational processing or modifications once they are stored in the vesicles ready to be released. Then they bind to receptors in the cell surface. Examples of these are your insulin, glucagon, and your adrenocorticotropic hormone from the pituitary gland. You also have the other pituitary hormones, which are glycoproteins in nature, the luteinizing hormone, the follicle-stimulating hormone, the thyroid-stimulating hormone, and the human chorionic gonadotropin. The next classification are the hormones which are amino acid derivatives. Particularly, they are derived from the amino acid tyrosine. All of these amino acid derivatives, particularly the catecholamines, would bind to receptors in the cell surface since they are polar in nature, except for the thyroid hormones, which bind to nuclear receptors. The half-life can range from 2 to 3 minutes, such as your catecholamines, epinephrine, norepinephrine, while your thyroid hormones can last for 0.75 to 6.7 days. As I mentioned, examples of your catecholamines are norepinephrine, epinephrine, and dopamine. The thyroid hormones T4 and T3 will be discussed in the third video presentation. The last classification are the steroid hormones, and they are all derived from the lipid-soluble compound cholesterol. They are synthesized in the adrenal cortex, the gonads, and the placenta. And as such, they require binding proteins to circulate in the plasma. So you'll expect that you have corticosteroid binding globulins, okay, where glucocorticoids would bind to. As being lipophilic in nature, they readily cross the plasma membrane to bind to intracellular, cytosolic, or nuclear receptors. And the half-life can range from as little as 4 to as much as 170 minutes. Examples of these are cortisol and cortisone, aldosterone, testosterone, estrogen, and progesterone, which have been discussed in the previous module, in the previous bimonthly. Calcidiol and calcitriol, or vitamin D, were already discussed in the first bimonthly. A very important principle in endocrine physiology is the negative feedback mechanism. So you call this your endocrine axis-driven negative feedback. and there can be several levels of feedback. Okay, so just keep in mind that as an organ produces a particular hormone, it causes a negative feedback on organs which directly regulate the first organ. Okay, so for example, the first type of level is the long loop, wherein the hormone, once it is produced, can cause a negative feedback on the hypothalamus. Okay, and then the short loop is when your tropic hormone causes a negative feedback on the hypothalamic neuroendocrine neurons. And it's ultra short loop if the releasing hormone itself coming from the hypothalamus would cause a negative feedback on the hypothalamus itself. So keep in mind that you have these three types or levels of feedback: long loop, short loop, and ultra short loop. The hormone is synthesized and stored in vesicles for some, then it can be ready for transport. So they are released into the circulation and can either circulate freely or bound to a particular protein called your carrier proteins or your binding proteins. These binding proteins serve as a reservoir for the hormone and will increase the hormone's half-life. So keep in mind that the half-life is the time during which the concentration of a hormone decreases to 50% of its initial concentration. So the more the hormone is bound to a protein, the longer the half-life. So in other words, the free or unbound hormone is the active form of the hormone, which binds to the specific hormone receptor. So remember that. Okay, so the hormone binding to its carrier protein serves to regulate the activity of the hormone by determining how much hormone is free to exert a biologic action. So hormones can also be classified as either free or bound. Examples of your free hormones are your amines and your peptides, while the bound hormones are the steroids and the thyroid hormones. So steroids bind to your CBG or your corticosteroid binding globulin, while thyroid hormones would bind to TBG or your thyroid binding globulins. The half-life of a particular hormone can also be influenced by its rate of metabolism. So in this slide, it is inversely related to its removal from the circulation, or what we call as your metabolic clearance rate, which is the volume of plasma cleared of the hormone per unit of time. And once the hormones are released, they either bind to their specific receptor in a target organ, or they may undergo metabolic transformation by the liver, or lastly, undergo urinary excretion. Once they reach the liver, hormones can be metabolized in these various reactions. So you have phase 1 reactions such as hydroxylation and oxidation, and phase 2 reactions such as glucuronidation, sulfation, or reduction with glutathione. These reactions will serve as a mechanism for clearing the body from the hormones, okay, before they can be excreted either in the feces or in the urine. This figure summarizes the various factors that determine the blood concentration of hormones. So the level of hormone in the blood can increase if you have increase in the synthesis of these hormones, or in the increase in the availability of your chemical precursors such as cholesterol or tyrosine. Okay, but they can also be decreased if you have increased uptake in the target cells or increased excretion by the liver or the kidney. So this is basically how these factors affect concentrations of hormones in the blood. Okay, before I end this video presentation, make sure that you're able to read chapter 75 for this first lecture. Especially, you have to study table 75-1 for a summary of the endocrine glands, the hormones, and their functions and their structure. These are my references, and see you on my next video presentation.